This video we're going to talk about protein structure and how that structure is related to the function of the protein. Now, when we talk about protein structure, we need to reference 4 levels in a hierarchy, and we always start with the primary structure. Now, that is quite simply the number and sequence of amino acids, which are the subunits of a polypeptide chain. The secondary structure we're going to refer to as the folding of the primary structure. Now, that secondary structure can again be folded into a more complex 3D shape, and we refer to that as the tertiary structure. Now, some proteins might have a quaternary structure, and this is where we get 2 or more polypeptide chains that interact together to form a functional protein. Now, we can classify proteins into 2 main groups. The first is the globular proteins. Now, these might have functions that include channel or carrier proteins that we find in phospholipid bilayers. They might be enzymes that catalyze biological reactions. They might be antibodies which bind to certain antigen molecules, maybe on the surface of a bacterial cell or a virus. And we can have globular proteins that form hormones that will bind to receptor molecules on the surface of some cells. Now, fibrous proteins have a different structure, and we're going to talk about collagen and keratin, and they can help form what we call connective tissue. So, for example, collagen will form connective tissue within the skin. It can also be tendons, ligaments, and cartilage within the human body. Now, this diagram represents the primary structure of a polypeptide chain, and if we look at the subunit on the very right hand side, we can see this red circle has the letters ASN inside it. Now, that's just shorthand for that particular type of amino acid called asparagine, and that will be bonded to the second amino acid in the chain. GLY in this case references glycine. Very similarly, we've got the third amino acid, this green circle. PHE represents phenylalanine. Now, you don't have to know the names of the amino acids, they'll be given in the exam question, but you do need to know that the primary structure is the total number and the sequence of the specific amino acids that make up the polypeptide chain. Now, if we look how these amino acids are bonded together, the first amino acid, ASN, will bond to GLY, number 2, by a peptide bond. And the second amino acid, likewise, will bond to the third amino acid by another peptide bond. And that's a C to N bond that we'll do very shortly. So if we go all the way down the polypeptide chain, we're going to find a whole series of peptide bonds that link each of the amino acids together that make up the primary structure. Now, each amino acid is actually coded for from the genetic material that we call DNA. And there's 3 bases that form what we call a codon. Now, that triplet code specifies that specific amino acid. So a gene is a sequence of codons that specify the sequence of amino acids in the polypeptide chain. Now, the primary structure determines the overall structure of the protein and therefore the overall function of that particular protein. Now, this is the basic generalized structure of an amino acid, and we have to be able to draw this and be able to recognize it. So if we have a look on the left hand side, what we can see is the amine group, and that has nitrogen within it, a nitrogen atom. Now, bonded to that nitrogen atom, we've got 2 hydrogen atoms, and we can refer to this as the NH2 or the amine group. Now, proteins are a bit different to carbohydrates and lipids because they do contain the element nitrogen. Now, if we have a look on the right hand side, we can see a different type of group. This time it's called the carboxyl group, and we have a carbon atom, and that's double bonded to an oxygen atom, and then it's also bonded to another oxygen that will form what we call the hydroxyl group. So OH represents a polar hydroxyl group, and we can reference this as COOH, carboxyl group. Now, in the middle, we've got a carbon atom. It's called the alpha carbon atom, and that is bonded covalently to a hydrogen atom. Now, on the other side, we've got what's called an R group. Now, because we've got 20 amino acids in nature, we're actually going to have 20 different variable groups. And the variable group determines the properties of that particular amino acid. It might be a hydrophilic variable group, it could be a hydrophobic variable group. So we can start to draw individual amino acids as long as we're provided with the variable group. So if we look at glycine in yellow at the top, it says the R group is a hydrogen atom. So if we look in green underneath, we can actually see we've got a generalized amino acid structure with the amine group on the left and the COOH group on the right. The central carbon atom is bonded to a hydrogen, and this time the variable group is a hydrogen atom that we can just draw in here. So we're going to replace the R with a hydrogen atom. Now if we look at alanine, the next one, it's exactly the same generalized structure, but this time the R group that we're provided with is a methyl group. Now if we look at serine, this is a very, very similar scenario. However, this time the variable group is CH2OH. So we draw the basic amino acid structure with the amine group on the left hand side. So I'll just quickly highlight that. The COOH group on the right hand side, that's the carboxyl group. The central carbon atom and the hydrogen above. And we just put in CH2OH. Now we know OH groups are polar. And that means that the oxygen has a very partial negative charge, or a small negative charge, and the hydrogen atom in the hydroxyl group has a very small or partial positive charge. And because water is also a polar molecule, it means it's going to interact and form hydrogen bonds with this hydroxyl group, and that's going to make serine fairly soluble in water. Now lysine is another amino acid, and here if we just highlight the variable group, we've got CH2, but this time 4 times. So there you go. And on the end, we've got an NH2 group. So all we do, again, is we just draw in the generalized amino acid with NH2 on the left, with COOH on the right-hand side. In the middle, we've got the carbon atom. At the top, we've got the hydrogen. And below, we've got the variable group. Now, it doesn't really matter which orientation you draw the hydrogen atom and the variable group. So sometimes you might see the variable group popping up from the top, and sometimes the hydrogen atom will pop down below. Now the extension question says, which is the polar variable group that is going to be hydrophilic? Well, because we know OH groups or hydroxyl groups are polar, they're going to form hydrogen bonds with water molecules. So we're going to say serine is the hydrophilic variable group. Now the nature of the variable group determines the nature of the protein itself. The next thing we have to be able to draw is a dipeptide. And this is quite simply 2 amino acids linked together through what we call a peptide bond. So we can see on the left hand side we've got a generalized amino acid. So we've got our NH2 group on the left, COOH group on the right hand side, a central carbon atom and a hydrogen atom at the top, and it's exactly the same for the amino acid on the right hand side. Now in order to draw a peptide bond, what would happen is an enzyme would remove a molecule of water, and we're going to call this a condensation reaction. So the carbon atom on the left hand side, the carboxyl group on the left hand side, that is now going to link with, just circle that, the nitrogen atom of the amine group from the right hand amino acid. And to do this, the OH group on the left hand side and the hydrogen atom bonded to the nitrogen on the right hand side are going to be removed, and the covalent bond that forms is called the peptide bond, and that's always between the carbon of the left hand amino acid and the nitrogen of the right hand amino acid. Now this reaction is reversible, and as you might work out, the chemical addition of water by an enzyme is called hydrolysis. So many bonds can be broken by adding water through an enzyme catalyzed reaction. Now if we look at the 2 amino acids on the bottom, we can see the OH group has been re-established, bonded to the carbon of the carboxyl group, and the hydrogen atom is bonded to the nitrogen of the amine group on the right hand side. So we end up with the 2 amino acids that we originally started with. So condensation is the removal of water by enzymes to form a covalent bond. In this case it's the C to N in the middle. That's our peptide bond. And below, hydrolysis is an enzyme reaction that adds water to break apart a covalent bond, in this case the peptide bond. Now the diagram in the middle, the peptide, dipeptide, is the one that you have to learn, but always do remember that that is one of the products. from the two amino acids joining together. The other product that you always have to include is water. So quite often you'll get 2 marks for this diagram, one for the dipeptide and one for the water that's been taken out. Now, some properties of amino acids then. This is the way we draw it in our textbooks. So we can see the amine group on the left and the COOH carboxyl group on the right hand side. We've got a central carbon atom. This time the hydrogen's just been drawn next to it. We've got our variable group beneath the central carbon atom. However, what we do know is that in that solution, the amine group on the left hand side, it can act as a base, and that means it can actually bind protons or hydrogen ions. Now the carboxyl group COOH on the right hand side, that can act as an acid, so that can actually donate a proton. So sometimes we'll actually see the amine group drawn as NH3+ because it accepts protons and acts as a base, and sometimes we see the carboxyl group drawn as COO- because it can donate protons and therefore act as an acid, and that is called amphoteric. Now we're going to talk about the primary structure of a protein. So we can now start to draw a polypeptide primary structure. Now if we look at the diagram at the top, we can actually see that there's 8 amino acids that are linked by peptide bonds to form quite a short polypeptide. On the left hand side we've got the amine group NH3+, and on the right side we've got the carboxyl group COO-. Now we will need a key to be able to draw the polypeptide because this details the variable groups. So glycine on the top row at the left hand side we can see is a hydrogen atom, and alanine, amino acid number 2, is a methyl group, and they are both non-polar. That means they're not going to interact with water. Now amino acid number 3 is serine, and that's in the middle row here, so we can see CH2OH. Now we know OH groups are polar because we know the oxygen is electronegative, so the oxygen has a small negative charge, the hydrogen has a very small or partial positive charge, and that hydroxyl group with it will interact with water molecules that will form hydrogen bonds. Now if we have a look further down the polypeptide chain at the top of the page, we can see phenylalanine, that's amino acid number 5. Now that's in the top row, and that has CH2 and then a benzene ring, and a benzene ring is just made from carbon and hydrogen, and it's actually non-polar, and it's actually very hydrophobic. Now that means this particular variable group, and therefore this part of the polypeptide chain, will repel water. Now cytosine is amino acid number 8 in the polypeptide chain, and that's also quite interesting because if we have a look at the variable group, it's CH2SH, and this is the middle row. Now it's got sulfur in the variable group, so we know cysteine amino acids can actually form what we call disulfide bonds, and they're quite strong covalent bonds that help maintain the tertiary structure of a protein that we'll see a little bit later. Now because this polypeptide has 8 amino acids, it indicates to us that we're going to be able to draw in 7 peptide bonds. So the length of the polypeptide chain, if it was say 20 amino acids, it would have 1 less peptide bond, so that would be 19 peptide bonds. If we had 30 amino acids, we would have 1 less, which would be 29 peptide bonds. Now if we draw this polypeptide out nice and neatly, what we can see is in grey are the 8 amino acids. We can see the carbon atom here, and that is covalently bonded to the nitrogen, and that's our peptide bond, and we're going to get 7 of these if we just count them out. So we can see 7 peptide bonds, C to N. We're going to have the amine group on the left hand side and the carboxyl group on the very right hand side of the polypeptide chain. Now this is the primary structure again, and we can see it's the number and sequence of specific amino acids along the length of the polypeptide chain. Now sometimes if you get a mutation in DNA and that changes a codon, it might actually change the amino acid that is specified at that particular position along the gene. Now, sometimes if an amino acid changes in the length of the polypeptide chain, if that amino acid is involved in forming one of the 4 bonds to help the protein finalize its shape, it can actually affect the function of that protein. That's why sometimes we see mutations in DNA are actually translated into a change in the polypeptide chain, which has a different structure and therefore might not perform its job. Now, normally these mutations will only have an effect if that specific amino acid is involved in the coiling or the final shape of the protein. Now, we're going to talk about secondary structure next. So there's 2 main types of secondary structure. The first one is the alpha helix, and this is the more common of the 2. Now, if we look at the polypeptide chain, we can actually see it's in a spiral shape, and we call that the alpha helix. Now, the reason that this polypeptide chain will take such a shape is because of hydrogen bonds. Now, if we look at the carbon to nitrogen here on the left hand side, this is the peptide bond at one position in the chain. We know the carbon atom is double bonded to an oxygen. Now, if we then go further down the polypeptide chain, we will see another peptide bond. So here we know it's C to N, and we know the nitrogen is bonded to a hydrogen atom here in blue in the middle. So what we do see is that between the oxygen bonded to the carbon and the hydrogen bonded to the nitrogen, we're going to get a weak intermolecular bond, and that's called the hydrogen bond. Okay. Now, that on itself is quite weak, but when we've got many of them within this secondary structure, it's quite collectively a strong force, and it maintains it in a very stable alpha helical structure. Now, the other type is the beta pleated sheet. Now, here there's no coiling or spiral shape, but instead the polypeptide chain, if we look at the backbone, we can actually see the backbone's in a type of zigzag shape. The polypeptide chain then coils round, and we might get another portion of the polypeptide chain that takes the zigzag shape. And in this particular case, we've got another one here, so that's going to form another zigzag shape, and then we'll get to the end of the polypeptide chain. So if we actually look at some of the atoms here, we can see a carbon atom in the middle in blue, bonded to the nitrogen. That's the peptide bond. Now, we know the carbon, just like above, is attached to an oxygen atom. Now, if we go further down the polypeptide chain, we're going to see another peptide bond, okay, here in blue in the middle row. And we know the nitrogen is bonded to a hydrogen, so we're going to see between the oxygen and the hydrogen a hydrogen bond. And we know if we've got multiple hydrogen bonds, that's going to hold this zigzag shape, this secondary structure, in a nice stable configuration. Next up, we've got the tertiary structure of a protein. So if I draw in the alpha helix, for example, the secondary structure, imagine this purple line was an alpha helix. Okay, so a nice spiral shape. Now, the tertiary structure is actually the 3D folding of the alpha helix into a more complex globular shape. So there's the alpha helix. Now, if we have a look at different amino acids along the length of the alpha helix, imagine we start here. So this amino acid at this position on the polypeptide chain. The variable group for this amino acid I've just outlined in blue. Now, if we keep going down the polypeptide chain, we will get to another amino acid further along. So imagine this one in blue down here. Now, the variable group of this particular amino acid has the ability to hydrogen bond with the variable group of the initial amino acid in blue that we talked about earlier. And we will call this a hydrogen bond. And we know that's quite a weak interaction, but it's very important to maintain this complex 3D shape. Now, the other 3 bonds that we're going to see are called ionic, hydrophobic interactions, and disulfide bonds. So if we start with the ionic bonds, then we can look down here. So if we've got an amino acid further down the polypeptide chain, this variable group here, it has 4 CH2 groups and an NH3 plus at the end. So that's got a positive charge. Now, if we keep going down the polypeptide chain, we will get to another amino acid at this position, And the variable group this time that pops out is CH2COO-. So that variable group has a negative charge. Now we know a positive charge will attract a full negative charge, and that's called an ionic bond. And again, that's a fairly weak intermolecular interaction. Now, the hydrophobic interactions then, if we look up here in pink, this box here, imagine you had an amino acid in orange there. Now, the variable group here is the one that I've just circled, CH3, and that's hydrophobic because it's got no polar groups and it's not charged. Now, further along here, we'll get to another amino acid which has exactly the same variable group. Now, because it's the same variable group, it actually makes it exactly the same type of amino acid. And hydrophobic variable groups will tend to cluster together because they will sit energetically just next to each other. Now, the last type of bond is the disulfide bridge. So imagine we had an amino acid at this point in black along the chain. Now, the variable group here is CH2SH. So SH at the end. Now, if you keep going down the polypeptide chain, you will eventually get to another amino acid, which is cysteine, so it's the same, with the same variable group. So that would actually finish in SH as well. Now, what can happen between these two cysteine variable groups is that hydrogen is removed through what we call an oxidation reaction. So it causes the formation of a covalent bond between the two sulfur atoms, and that's why we call it a disulfide bridge or disulfide bond, because it's between two sulfur atoms. Now, this is a covalent bond, and it's really important in being able to maintain the 3D shape of that particular tertiary structure, and it's quite a strong covalent bond. The others are slightly weaker intermolecular forces. Okay, the last shape is the quaternary protein. Now, we can define this as two or more polypeptide chains in a tertiary structure normally that can interact to form a functioning globular protein. So the example here for the globular proteins is what we call hemoglobin, and we've got 4 subunits. So we've got 4 separate polypeptide chains. So 1, 2, 3, and 4. Now, this chain here is called a beta chain. It's nothing to do with a beta pleated sheet. It's just a name given to the subunit. Now, the subunit in pink down here, number 3, is actually the same or identical to subunit number 1. Subunit number 2 up here on the right hand side, we're going to call that an alpha subunit, and that's exactly the same as subunit number 4. So there's 2 types of subunits in hemoglobin, and there's 2 of each. So we've got 4 polypeptide chains, and each one is in what we call the tertiary structure. So we've got 4 tertiary structures that are going to bond and interact together to form the functional protein. Now, the 4 bonds in the quaternary structure, they're exactly the same as the 4 that we mentioned for tertiary. However, the big difference here is that they occur between variable groups of different polypeptide chains. So we might have a variable group from subunit number 1 at a certain position, and it might interact with another variable group from an amino acid as part of subunit number 2. Now, the other thing we need to talk about with hemoglobin is we do find what were called prosthetic groups. So here, if I circle these, we're going to call these prosthetic groups. And that means it's a non-protein group attached to the protein part of the molecule. Now, in this particular case, in hemoglobin, there's an Fe2+ ion as part of the prosthetic group, and that's the part of the prosthetic group that binds oxygen. So hemoglobin can actually bind 4 oxygen molecules at any one time. Now, we're going to talk about globular proteins and their function. So one role of globular proteins is as carrier or channel proteins that we find that span phospholipid bilayers. So the word intrinsic actually means spanning the entire phospholipid bilayer. Now, this channel protein here is actually a chloride ion transporter, and it will allow chloride ions to go from high concentration through a central hydrophilic pore to a low concentration of chloride ions. Now, if we look at how this particular protein inserts into the phospholipid bilayer, what we might expect is the variable groups part of the protein that face the fatty acid tails, so maybe here. And we know fatty acid tails are hydrophobic. So the variable groups in this portion of the channel protein on the outside of it must also have hydrophobic variable groups. That's because we know hydrophobic molecules tend to sit next to each other, which will allow the channel protein to sit quite happily within the membrane and span the membrane structure. Now, if we look at some of the variable groups maybe on the outside of the polypeptide chain, so imagine these amino acids here had variable groups. Now, these variable groups must be either polar or ionic, and that's because the tissue fluid that we find outside the cells of our body contains a lot of water. So variable groups have to be able to interact with this. They have to be hydrophilic. And it's the same with the variable groups that we're going to get along the length of the polypeptide chain that faces the cytoplasm of the cell. Because again, they have to be hydrophilic and they have to be able to interact with the cytoplasm, which contains a lot of water. So the variable groups as part of the tertiary structure are going to directly determine how this protein fits into a membrane. Now, the inside of the protein has a hydrophilic pore, so the variable groups that project into this pore will also be hydrophilic, because we know there's a little stream of water that passes through the pore, and that allows dissolved chloride ions to go from the tissue fluid by facilitated diffusion across the channel protein and into the cell. Now, another example of a globular protein is an enzyme. And enzymes are all tertiary proteins, and that means they're made from a single polypeptide chain that's been folded into a complex 3D shape. Now, the shape of the enzyme is really important because it gives the enzyme a specifically shaped active site into which the substrate will fit. Now, this particular enzyme catalyzes the breakdown of hydrogen peroxide, which is a waste product of metabolism inside all cells, into water and oxygen. Now, hydrogen peroxide, because it's a waste product of metabolism, if it accumulates, it's actually quite toxic to the cell. So cells that make a lot of hydrogen peroxide will actually express a lot of catalase, and that allows the hydrogen peroxide to be broken down into water and oxygen. And these products are not toxic to the cell. Now, why is the tertiary structure of a protein important for its function? Well, at A level we do a thing called the induced fit model, and it's the mechanism of how this protein works, this enzyme. So if I highlight the shape of the active site, we can see it's complementary in shape to the shape of the substrate. But the induced fit model suggests that the complementarity is not 100%, but initially there's going to be a loose fit of substrate to active site. Now, once the substrate has bound to the active site, if it's the correct substrate and there's the correct interactions between the variable groups of the substrate and the active site, we get an induced change to the shape of the active site, and then it perfectly fits around the shape of the substrate. And when that occurs, we then get the enzyme substrate complex. Here the substrate can then be twisted, some of the bonds are broken and new bonds will be formed. That will form a brand new product or products, and they will, because they've got a different shape to the original substrate, they'll be released from the active site. Now, the important point here in the induced fit model is that the shape of the active site goes back to its original shape, and that means the enzyme is going to be reused and it will bind another substrate molecule and it will catalyze exactly the same type of reaction. Now, another type of globular protein is the antibodies, and they have a quaternary structure. So if we look at this diagram on the right hand side, we can see there's actually 1, 2, 3, 4 polypeptide chains, so we know it must be quaternary. Now, the first chain, number 1 here, if we have a look carefully at that structure, we can see it's a lot shorter than chain number 2. So we're going to call this the light chain. And if you look at chain number 4, it's virtually identical. So chains 1 and 4 are called the light chains. Chain 2 and chain 3, as you can probably see, they're a lot longer in length, so we're going to call these the heavy chains. So that's chain 2 and 3. Now at the tips of chains 1 and 2, we're going to get a very specific shape, and that's exactly the same as the shape that we get at the end of tips 3 and 4, and we call this the hypervariable region. Now each individual antibody quaternary protein will bind to a specifically shaped antigen, and an antigen can be a molecule on the surface of a bacterial cell or a virus cell. So we're going to get this complementarity, and we're going to get binding between the tips of the antigen, the tips of the antibody and the antigen molecule itself. Now when antigens bind to the antibody, it's the antibody that will then stimulate further immune reactions. And so quite often these pathogens, if they're bacteria or fungi, etc., will get destroyed by other immune cells that then are brought to the site of infection. Now on the left hand side you can actually see this globular protein. It's quaternary, there's 4 chains. Now this part here in blue, this is the heavy chain, and at the end here with the light chain in green, we're going to get the hypervariable region, and that's complementary in shape and specific to a certain antigen molecule. Now the fibrous proteins are next. So we know fibrous proteins include both keratin and collagen. Now the structure is very different to the globular proteins. So in collagen, for example, we have 3 polypeptide chains, and each chain has an alpha helix secondary structure. So we've got 3 alpha helices that come together to form a more complex quaternary structure. There's no tertiary structure in collagen, but we have 3 elongated alpha helical secondary structures. Now the amino acid sequence along each of these polypeptide chains tends to be repeated, or there are certainly repeating elements to the polypeptide chain. That's another characteristic of the fibrous proteins. Now the chains may be held together by hydrogen bonds. So the polypeptide chains will have lots and lots of hydrogen bonds that help to hold the 3 alpha helices together, and we call this fiber a tropocollagen fiber. Now we know fibrous proteins are insoluble, so both collagen and keratin are insoluble. They are elongated, they have repeating amino acid sequences down each polypeptide chain. There's no tertiary structure, and they're going to form part of connective tissue. So collagen is found in the skin, the connective tissue of the skin, cartilage, tendons and ligaments, and we find keratin in slightly harder structures called nails and also within the hair.